Hello everyone. So in today's videos, I will be talking about the biochemical basis of the treatment of urea cycle defect. And last video I had talked about, in great detail about the laboratory aspect related to urea cycle. So today we'll be looking into the treatment part. So how, or we can say how we can approach with the patient which is already diagnosed with urea cycle defect. So what could be the line of the treatment? So the today videos or in today lecture we'll look into the treatment part of urea cycle defect. So the very first line of treatment should be complete restriction of protein based diet or protein containing diet. Okay, so there could be complete restriction of protein containing diet. Why that is so? Since you know that protein are catabolized in our body to give what? Ammonia, and this ammonia with the help of urea cycle is converted into urea, so which is happening in liver. So here, due to defect in the urea cycle enzyme or due to defect in the transporter which help in urea cycle, so that can lead to increased formation of ammonia, because since this ammonia is not getting converted into urea due to what? Due to defect in the urea cycle enzyme in this individual or in this patient. So ammonia level will increase, and if the ammonia level increases, that is called hyperammonemia. So this ammonia can cross blood-brain barrier, okay, and this blood-brain barrier, after crossing this blood-brain barrier, this ammonia can be highly toxic, okay, and it can lead to severe neurological symptoms including coma and death if it is not treated properly at right time. So the very first thing which we should do once the patient is diagnosed with urea cycle defect, whether it is acute case or chronic case, there should be complete restriction of protein containing diet. That is the first line of treatment. So there will be less load of ammonia or less formation of ammonia, and obviously the ammonia toxicity will be less. That is the first line of treatment. The second line is, in order to decrease catabolism, because there could be the chance that protein catabolism inside body might happen, and this excess catabolism of protein, again it will produce what? Ammonia, and ammonia, since urea cycle is defective, that ammonia cannot be processed by our liver. So obviously, so in order to reduce catabolism, we should give intra-IV administration, intravenous administration of 10% dextrose or glucose is given, so that the high calorie rich glucose will serve as a source of energy, so that the catabolic rate will be low, and because this glucose will take care of the energy requirement of the body and the protein will be spared. So that's why, in order to avoid protein degradation or catabolism inside our body, we should give intravenous 10% dextrose to urea cycle defect patient, or where there is a deficiency of urea cycle or already diagnosed cases of urea cycle defect, so that in order to decrease or avoid catabolism or especially protein catabolism in our body. So that is the second line of treatment. And of course we can give, there are other line of treatment which are given with this 10% dextrose. So what we can do is, we can give certain, we can go for acylation therapy. What does acylation therapy do? So what we do is, along with in 10% dextrose solution that is given through intravenous route, we can also give phenyl. We can go for sodium benzoate. Is given. So let's look into the mechanism of sodium benzoate. So this is given along with 10% dextrose. Okay. So sodium benzoate, what does it do? How it eliminates ammonia? So here you should remember one thing, that in urea cycle defect, what we should take care of is ammonia level, blood ammonia level. So blood ammonia level shouldn't go high. If it is on higher side, we have to lower it by giving certain treatment. So what could be the probability of the treatment? So there could be two ways by which ammonia can be reduced. The first one is by restricting protein based diet which have higher content of protein, or with the diet which is rich in protein, that we can restrict. The second thing what we can do is we can eliminate ammonia through urine. There could be, we can increase the excretion of ammonia through urine, so that blood ammonia level will go down or it will come down. So what we can do is we can give certain medication through intravenous route, okay, along with 10% dextrose. So the first medication which is very effective is sodium benzoate. Okay, what does it do? What is going, once this sodium benzoate, once it is administered through IV route, what it's going to do? What is the mechanism? So this benzoic acid inside our body combined with coenzyme A to form what? Benzoyl CoA, and this benzoyl CoA will react with a non-essential amino acid like glycine to form what? Hippuric acid. And this hippuric acid can be easily excreted through urine. So indirectly what this benzoic acid is doing is it is eliminating ammonia in the form of glycine, because glycine is also one of the sources of ammonia, because it is a non-essential amino acid. It can produce ammonia through process called transamination and deamination. So of course, by reacting with glycine, this benzoyl CoA forms a excretable product called hippuric acid, which can be easily eliminated through urine. So it is said that 1 mole of benzoic acid removes 1 mole of ammonia in the form of glycine. So the ammonia level in blood will go down. So that is the first drug of choice which can be given to increase excretion of ammonia in the form of glycine, because glycine is also one of the source of ammonia, and this glycine can bind with benzoyl CoA to form what? Hippuric acid. So that is the first drug of choice. The second drug of choice which can be given, that is also part of alkylation therapy, that is sodium phenyl acetate. So what does it do? Once it is administered through intravenous route, this sodium phenyl acetate is going to combine with acetyl CoA to form what? Phenyl acetyl CoA. It forms what? Phenyl acetyl CoA inside our body, and this phenyl acetyl CoA will bind with another non-essential amino acid, that is glutamine to form what? Phenyl acetyl glutamine. And here we are excreting ammonia, and this phenyl acetyl glutamine will be excreted through urine. So here we are excreting ammonia in the form of glutamine. Okay, so glutamine is also one of the source of ammonia. Okay, and this glutamine is binding with this compound called phenyl acetate to form what? Phenyl acetyl glutamine in our body, and this is easily excretable through urine. So it is said that 1 mole of sodium phenyl acetate removes 2 mole of ammonia in the form of phenyl acetyl glutamine. So that is also one of the important line of treatment which could be given to to reduce the level of ammonia in blood. The third line of treatment which is also very effective is arginine. That is one of the, it comes under semi-essential amino acid, arginine. Arginine can be given through intravenous route along with 10% dextrose. So this arginine is given in the form of arginine hydrochloride. Okay, and this arginine, what it's going to do? Arginine, you know that it is an activator of one enzyme called N-acetyl glutamine synthase. Okay, so what does this N-acetyl glutamine synthase do? Basically, what does it do? This acetyl CoA and glutamate, in the presence of enzyme N-acetyl glutamate synthase forms what? N-acetyl glutamate, and this N-acetyl glutamate synthase requires arginine as activator. Okay, N-acetyl glutamate synthase require arginine as an activator. Okay, So this N-acetyl glutamate synthase will form N-acetyl glutamate, and this N-acetyl glutamate is allosteric activator of enzyme called CPS1, that is the very first rate limiting enzyme of urea cycle. So what arginine is going to do? So since it is increasing the formation of N-acetyl glutamate, so this CPS enzyme will be stimulated and this ammonia will be processed at much higher rate, or this ammonia accumulation will be prevented, because CPS1 is the first enzyme where that CPS1 enzyme, if you remember the step, this CPS1 catalyze the reaction where the ammonium ion and carbon dioxide bind to form what? Carbomoyl phosphate. So if there is increased level of ammonia and if arginine is administered through intravenous route, so what will happen? This arginine will stimulate the enzyme called N-acetyl glutamate formation, okay, and this N-acetyl glutamate in turn will stimulate CPS1 and there will be more processing of this ammonium ion. So ammonia level in blood will decrease. So that is also one of the treatment. Only contradiction here is this arginine cannot be given in arginase deficiency patient, okay, which have arginase deficiency, that is also one of the urea cycle defect. So if the person has arginase deficiency, we cannot give arginine through intravenous route where it cannot work, okay, arginine won't work, but in all other types of urea cycle defect, arginine will serve as a as a source for eliminating ammonia or for reducing ammonia. So it can be given as a part of the treatment. Apart from that, this arginine is also an essential amino acid, because here this since urea cycle defect is there, this arginine will serve as a source of essential amino acid, and this can help in the synthesis of protein. So that also is very important in urea cycle defect patient, because this urea cycle defect, arginine cannot be synthesized by our body. So there it will be also a source for synthesis of protein. So there also arginine would be handy. But only drawback of arginine treatment is it cannot be given in in that urea cycle defect where there is a deficiency of arginase enzymes. There arginine will cause toxic effect. Okay, so that's where we shouldn't give arginine. Apart from that, arginine can serve as a very good medication for eliminating excess ammonia in the blood. So I hope the biochemical part or the biochemical basis of the treatment of urea cycle defect would become now after my lecture, that would have become more easier to understand, and keep liking and keep sharing my videos. Thank you.